It's a pleasure for me to introduce before you Divya Ambati ji, who is Agriculture Research Service Cadre Batch 2010. She is an ICAR scientist since September 2011. She is presently posted at Indian Institute of Oilseed Research, which is a prestigious institute of Indian Council of Agriculture Research. This institute is located at Hyderabad, Andhra Pradesh. She had come to Indore, and I thought it advisable to ask and request her to deliver a lecture for the IPS students, and she will be speaking on diseases of wheat and breeding strategies for rust resistance. As you may be knowing that wheat is an important crop, number one crop in India as far as the food consumption goes. Sometimes number one, sometimes number two, rice replace wheat, wheat replace rice, is like that. But it is estimated that wheat cultivation occurs in about 35 million hectares of land in India, and its production of 2020-21 had been 108 million ton of all the wheats, all the varieties which are grown in India. And it is estimated that the losses which occur because of the various diseases and various factors is about 5 to 50%. Sometimes it is 5%, sometimes it goes up to 50%. So it is very essential that breeding strategies should be adopted to develop varieties which are not only disease resistant but high yielding too. As you know, wheat, common wheat which we eat is a hexaploid wheat. It contains 42 chromosomes which are derived from 3 ancestral genomes, and the 21 pairs of the chromosomes can be further divided into 7 groups of 6 chromosomes. That Dr. Ambati will be discussing with you shortly. You will be surprised to know that it has fewer pairs of chromosomes than human beings, but it has greater number of genes, and the number of total genes which are estimated to be 1,64,000 to 3,34,000 genes, compared to about 25,000 genes present in the human being. So genetically, if you ask me, the wheat had got 3 genetic levels. One is the diploid species, another is the tetraploid species, and third is the hexaploid species. The common wheat which we consume is a hexaploid wheat which contains 42 chromosomes. And these 42 chromosomes can be easily found out through cytological studies in the somatic cells of the wheat species. Coming to the diseases, there are so many diseases which occurs in wheat like powdery mildew, loose smut, brown rust, yellow rust, black rust, flag smut, stinking smut, etc. Besides this, there are some few bacterial diseases also which occur on wheat, but the losses which are estimated every year in India are much more because of the rust diseases. So she will be mainly focusing on developing breeding strategies for rust resistance. As you might have read in your books about the life cycle of an organism Puccinia graminis. Its life cycle is complete on 2 different plants. One is wheat, another on berberis. So it's very important to know about the genetics of the diseases. And with these words, I hand over the stage to Miss Divya Ambati. She will be talking to you directly, and in between if you have any question, or after that you are free to ask questions.
First, I would like to thank Dr. Chan sir and Gupta sir for giving me this opportunity so that I can share my knowledge with you students. Just I would next 40 to 45 minutes I would like to talk about what are the important diseases of wheat and what are the breeding strategies that we adapt in All India coordinated programs for rust resistance, so that we can improve the wheat yield from 101.2 million tons, which was reported during 2019-20, and we need a total wheat production of around 140 million tons for the year 2050. So what are the strategies that we are following in our breeding programs that I am going to explain in next 40 to 45 minutes. First, I would like to explain the importance of wheat. As most of you know that wheat is globally an important crop for us, because after rice, wheat is the second most important crop which is cultivated in all over the world. After rice, after wheat, maize is the important crop, and it is an important source of protein for most of the population ranging from North Pole to South Pole, because this wheat accounts for 1/5 of our food. The food we consume, whether it is in the terms of in the product like roti, bread, biscuit, pasta, or vermicelli, or suji and dalia, in many forms we consume wheat. It accounts for around 1/5 of the total food we eat, and it gives around 22% of food calories, and it contributes to around 21% of the protein we consume every day. And in terms of production, India stands second to China in terms of production, and we produce around more than 100 million tons of wheat every year. And wheat also gains importance because it provides income to the farmers in more than 80 developing countries. And it is also an economically important crop because it is the most traded commodity in the world, and its export value ranges up to $50 billion every year. To the evolution of triticum species, earlier we had wild species which are diploids, and around 3 lakh to 5 lakh years ago, a natural hybridization between two diploid species, that is Triticum muratum and Aegilops peltoides, has led to the development of a tetraploid species which is called as wild emma or Triticum dicocoides. It was under cultivation, and around 10,000 years ago, because of the natural selection of the then farmers, it has led to the evolution of cultivated emma, which is called as Triticum dicocoides. Around 8,500 years ago, a natural mutation has occurred in this cultivated emma or Triticum dicocoides, and it has led to the development of spelta wheat, which is called as Triticum spelta. And a natural hybridization between this Triticum spelta and a diploid species called as Aegilops tauchi has led to the development or evolution of the hexaploid wheat called as bread wheat, which is scientifically known as Triticum istivum. And the natural selection of the farmers from this cultivated emma, which is called Triticum dicoccum, has led to the development of another tetraploid species called Triticum durum. So we have at present the wild species available, and also among them also we have diploid species, tetraploid species, and the hexaploid species. At present we have 3 cultivated wheat species around the world, and the most 3 important are the hexaploid species, which is called as common wheat or the bread wheat. It is having a chromosome number of 42 and scientifically known as Triticum istivum. And the second most important is durum wheat or a macaroni wheat, which is scientifically known as Triticum durum. It's an tetraploid species with a chromosome number of 28. And third most important is a dicoccum wheat or a hulled wheat, which is scientifically known as Triticum dicoccum, and it is also a tetraploid species with a chromosome number of 28. Coming to the most important wheat species, that is bread wheat. As I told, it's commonly known as Triticum istivum with a chromosome number of 42, an hexaploid species. It is the mostly cultivated wheat species all over the world. It occupies around 90% of total wheat area in the world, And coming to the production, it accounts for around 85% of total wheat production. And we utilize this bread wheat to prepare so many end products like chapati, bread, biscuits, etc. And it is having, in terms of quality, it is having good hectoliter weight. It means that within the same volume, it will accommodate so many seeds that it will be having more weight. These are few quality characters which we analyze, and we will decide on the basis of these quality characters, we decide whether this wheat, even the bread wheat also, the particular variety we have, whether it is suitable for making bread, or whether it is suitable for making chapatis, or whether it is suitable for making biscuits. Second most important is as durum wheat. It's a tetraploid species, and it is having a special importance in Indian context because it is suitable for making pasta. Along with pasta in India, we utilize it for making dalia, suji and vermicelli. But Indian durum wheat is having a good export potential for Middle East nations and the Mediterranean regions. And is also compared to bread wheat, durum wheat is assumed to be, it is having good nutritional characters, like it is having a good levels of protein content compared to bread wheat, and also the yellow pigment content, which includes beta carotene, which is a precursor of vitamin A, and also it's having a good micronutrient content like good amounts of iron and zinc. And also durum wheat is also known as hard wheat because it is more adapted to heat and drought areas compared to bread wheat. And also Indian durums, they have good tolerance to the diseases like loose mud, flax mud, and the canal bun diseases. Coming to the third important wheat species, that is dicocum wheat. It is a one tetraploid species which is being cultivated in the world from long ago. It is this plant have thin and long kernels, and it is useful for making breakfast foods like upmas. In South India, they prepare godi hugi, that which I have shown on the right hand side of the slide. It is mostly utilized in peninsular parts of India like Karnataka and Maharashtra. And it is also having outstanding grain qualities, and it is having good amounts of dietary fiber and protein. Because of its nutritional benefit, it is now considered as nutraceutical food, and it is mostly consumed by the patients who are diabetic because of presence of high fiber content. And it is also a good source of for resistance to various biotic and abiotic stresses, because the genes present in this dicocum wheat are different from the genes that are present in durum wheat and bread wheat. So this wheat can be utilized for further improvement of those two species. Coming to the Indian wheat production, during 1946-47, we were having an area of 9.84 million hectare, and production was only 6.5 million tons. After the inception of All India Coordinated Wheat Programs, and everyone has strategized how to improve the wheat production in India, and they have brought in how to improve the quality parameters and how we have to envisage our programs. And then in 1991, we have brought around 24.17 million hectares of area under wheat, and we could produce around 55.14 million tons. As I have explained in my first slide, for 2050, we have a target of around 140 million tons, which needed to be produced from this 35 million hectares, because the area is not going to increase anymore. At present, in 2021, there is an area of around 31 million hectare, and production of 108.75 million tons of wheat is produced. And the recent statistics for 21 and 22, it indicates that the area is around 35 million hectares, and production will be crossing 112 million tons. These are the different wheat growing zones of India. Like we have 5 main zones, like Northern Hill Zone, which includes Himachal, Jammu, Kashmir, and Northwest Plain Zone, which includes Punjab, Haryana, and Delhi. And we have a third important wheat growing zone, that is Northeast Plain Zone, which includes Uttar Pradesh, Bihar, Jharkhand, and few states of Northeastern Hill region. And we have a fourth important, that is a Central Zone, which includes Gujarat, Madhya Pradesh, Chhattisgarh. And we have a fifth wheat growing zone, a peninsular zone, which includes Maharashtra, Karnataka, and few parts of Tamil Nadu. And coming to the constraints we face when we go for increasing the wheat production, there are many constraints, but I would like to explain the biotic stresses. Mainly, those are being caused by fungal diseases. They are most important constraints, so that because of which we are facing around 5 to 50% of wheat losses every year. Coming to the northern hill zone, we have a problem with yellow rust and the powdery mildew. And in northwest plain zone, we have problems with yellow rust and kernel bunt. In northeastern hill zone, we have a problem with spot blotch and brown rust. In central zone, we have problems with stem rust and leaf rust. And in peninsular zone, we have problems with stem rust and leaf rust. The common, if you observe, the common problem among all these zones is rust. It's in northern part of India, we have problem with yellow rust and leaf rust. And in central and southern parts of India, we have problem with stem rust and leaf rust. So these are the main fungal diseases which are constraining the wheat production in India. We have wheat breeding priorities in India and all over the world. The core traits include we need to produce high yield, because that's the main factor which a farmer considers. Then you need to have a durable rust resistance, because rust is a problem all over the world where the wheat is grown. And also you need to have efficiency of these varieties for heat and drought tolerance. And also it is increasing the micronutrient content like iron and zinc content has also been given very much importance. And coming to the key diseases, like even though we have main criteria for improving yield, quality and everything, the problem comes again with the biotic stresses, particularly the wheat rust diseases. Coming to the important diseases of wheat in India and all over the world, the first three, as I have explained, the rust diseases are the most important. Apart from the rust diseases, we have flag smut disease that you can see on the top row towards the extreme right. That's a flag smut, in which the fungal spores you can see, like it forms a totally a black layer, and it affects the photosynthesis, and finally totally it affects the yield. And coming to the kernel bunt, the one on the top row to the left hand side, it's a seed-borne. It is a seed-borne disease, and it gives a stinking smell after the harvest of the wheat, and it creates a big problem for export of Indian wheat to the other nations, particularly to the European nations. And loose smut on down row towards the left, what you are seeing is a loose smut. During the formation of the spike itself, the fungus attacks and it totally converts the spike into a black powdery mass. It affects the total yield. As you can see, we won't get any seed where this disease affects. On the down row, second one, it's a powdery mildew. It's mostly seen in northern parts of India where we have cold conditions, and towards the southern hill zone, where in Nilagiri hills of Tamil Nadu, where the temperatures are low. Here it appears, the powdery mildew, it appears as a white powdery mass, and it affects the total photosynthetic area. It disturbs the total photosynthetic area and ultimately leads to the yield losses. And the down row, extreme left, it's a leaf blight. It's mostly observed in parts of peninsular India, that's in Karnataka and Maharashtra. The fungus affects the plant and creates a total black patches like this, and it affects the photosynthetic area, leading to the total yield losses. Coming to the most important diseases, which I am going to discuss, that's a wheat rust. As I have told, there are three important wheat rusts all over the world. The first one is black or stem rust, which is caused by the fungus Puccinia graminis tritici. And the second important is a brown or the leaf rust. It's scientifically known as Puccinia triticina. And third important is a yellow or the stripe rust. That's Puccinia stripiformis. Generally, the losses during the wheat rust range from 5 to 10%. But when there is an epidemic, like if we have a total leaf rust in that area or in total stem rust in that area, even the losses can go up to 50%. During 1890 to 1975, there were several rust epidemics were reported in India and on the other parts of the world. In the three pictures, on the left hand side, you can see the stem rest. It forms an, as the name indicates, it's stem rest. Stem rest, it means that it appears on mostly on the parts of the stem on the wheat plant. It can also be seen on leaf part and also on spikes also. It affects, it totally occupies covers the plant with these pustules and leads to the yield losses. And the second picture is a leaf rust. As the name indicates, it is having a brown color. It shows a brown color pustules and it appears only on the leaf. And it totally covers the leaf with the pustules during the growing season and leads to yield losses. And the third one you can see on the extreme right is the yellow rust or the stripe rust. It as the name indicates yellow, the pustules are yellow in color and appears in the form of a strip. Like you can see it in a linear manner, not in a vague way. It will be seen as in a linear manner on the leaves. So it is called as a stripe rust. As I have explained, this black rust is mostly seen in the areas where the temperatures are high, around 30 to 35 degrees centigrade. When the crop matures around 30 to 35 degrees, when the temperatures are around 30 to 35 degrees centigrade, then also you can see the stem rust. So this stem rust is mostly seen in hotter areas like peninsular India, that's in Karnataka and Maharashtra, and in central India like Madhya Pradesh, Chhattisgarh and Gujarat. Brown rust or the leaf rust, this is the most devastating rust diseases in the world, because it can accommodate it in any type of temperatures. Like it can accommodate in colder temperatures also, and also in the hotter environments. So all over India we have the problem with leaf rust. And stripe rust, it's a cool temperature loving rust species. So it's mainly seen in northern parts of India. Coming to the genus, the fungi which is causing this rust diseases, it belongs to the subphylum Puccinia, which consists of around 8,000 species of fungi, among which most of them are saprophytes. And they are parasites on plants, animals. And it belongs to the order Puccinales, which is the largest order with more than 90% of fungi belonging to the family Puccinia. Puccinia, the genus, it contains nearly 4,000 fungal species, and these rust species, they attack the plants by developing haustoria, and they absorb the nutrients from the host plant and they survive. This is the life cycle of Puccinia, which Chansar has explained you. The life cycle of Puccinia is also a very complex life cycle, as it includes both sexual and asexual phases of the fungi, and it produces 5 types of spores, which in the first one where you can see the basidiospores, they are the sexual spores which affect the secondary host or the alternate host called as barberry. It affects the barberry plant. It produces spermatogonia, and it leads to development of the spores called as you can see, the hyphae are produced. Then it leads to the development of achiospores. During this formation of basidiospores into achiospores, the sexual life cycle occurs, and during this there is a possibility of recombinations occurring, and it leads to development of new races of Puccinia. These achiospores infect the wheat plant. They complete their life cycle during the wheat growing season, and to survive the off season, they convert themselves into teleospores. These teleospores can survive any harsh type of conditions like cold or hot temperatures, and unless until they get their alternate host like barberry, they will survive. And once they get they get this barberry plant, they again attack. They convert themselves into basidiospores through a sexual cycle, and convert themselves into basidiospores and attack the barberry plant. Like this life cycle goes on, and wherever these alternate species like barberry is available, controlling this rust diseases is highly difficult, because these rust spores, they have 5 phases, 5 types of spores. Controlling each type of spore itself is difficult. Again, it is dispersed through wind. Even these spores can travel, any type of spore can travel lakhs of kilometers only because of wind. With the dispersal of wind, it can travel lakhs of kilometers together. So control, based on this life cycle itself, we can say how difficult it is to control this rust pathogens. And I would like to explain why these rusts are devastating. Because as I have explained, during the sexual life cycle, when they convert themselves from basidiospores to aciniospores, there the sexual cycle occurs, and they produce, because of recombinations, they produce virulent and aggressive pathotypes. And every year we get new types of pathotypes because of this sexual cycle and asexual cycles. And we have mainly three types of rust. And these new pathotypes, they are produced during the sexual life cycle, and they have alternate hosts also. Just as I have explained, this stem rust, for the stem rust, Barbary is the alternate host. Like that, for leaf rust, they have four alternate hosts. Like that, stem rust have different, stripe rust have different alternate hosts. So to have control all these alternate hosts is also a big problem when it comes to the for controlling rust diseases. However, during even asexual cycles also, these mutations can occur and lead to the development of new virulence. And as you know that farmers prefer the variety. And one more problem is the farmers prefer the variety which is new one and which is giving higher yield. They don't bother whether it is having rust susceptibility or something. They are least bothered. And we can see in all over India, 5 varieties, 10 varieties, they will be occupying total 35 million hectares of area. The contribution of other varieties is very less, because farmer is interested in particular varieties which are having good yield and good price. So the practice of this monoculture, because the varieties will be having resistance to only particular races of pathogen, they will be susceptible to the other pathogens. So this helps the pathogen to further multiply on the susceptible varieties when there is practice of monoculture among the farmers. Coming to the rust management, we need to follow the integrated method of disease management to control this rust. Among this, we have cultural methods, chemical methods. Cultural methods like we can alter the sowing time so that the crop escapes the pathogen. And irrigation, we can schedule the irrigation accordingly so that the plant does not provide so much nutrients to the pathogen to multiply. And also you can schedule fertilization and early maturing cultivars. Like we can sow the variety on the particular date, but it should get matured early so that it can escape the pathogen. And the chemical control is like you can use the fungicides, but as a scientist, I should not suggest a chemical control, because you know it's how environmental unfriendly is the chemical control. And coming to the third important rust management method on which we focus is host resistance. That we will try to develop the varieties which are resistant to the rust, so that you need not go for early sowing, or you need not alter the fertilization or irrigation facilities, and will be no need to go for a chemical control. This is for this you need to know what are the resistant genes that are available already in the farmers' fields and how much diverse they are. Because if farmers are cultivating 3 or 4 types of varieties, then we should be knowing whether the varieties are having same resistant genes or are they different. Even if a pathogen attacks, if the 4 varieties will be wiped off at a stretch, are there any varieties which can sustain this pathogen. And achievement of durable resistance, that is even though the pathogen changes its pattern, like even if it produces a virulence pathogens also, you need to produce a variety which can withstand all the new races. And deployment of resistance genes, like as I have told, we need to cultivate the varieties which are having different types of resistant genes. That's we call it as a deployment of resistant genes, so that the varieties which are being grown in central India are having different resistant genes compared to the varieties which are grown in peninsular India, and also the same like northwestern region and the northeastern region. How we breed for rust resistance? This breeding for rust resistance is based on the floor hypothesis, which was proposed in 1971. In simple terms, I would like to explain. It is a host pathogen interaction. When a pathogen comes and sits on the host, the host should have a particular gene which can withstand the protein that is produced by a pathogen. Pathogen is having a capital AVR gene, and the host pathogen and the host plant, like if it's in case of wheat, if it is having a capital R1 gene, if these two genes interact, like if the pathogen produces capital AVR and host is having capital R gene, then we don't have a disease. And the other three chances which are possible in the checkerboard as I have shown, if the pathogen is having small AVR and pathogen and host plant like wheat is having small r, then also we can expect a disease. Like this is a checkerboard which we follow, and based on the floor hypothesis, that's gene to gene interaction. The one gene is present in pathogen, that's a rust, and the other gene is present in the host plant, that's a wheat. The interaction between these two genes will lead to a compatible reaction or non-compatible reaction, and will lead to the progression of the disease. And at present, we have identified 79 leaf rust resistant genes, and 61 stem rust resistant genes have been identified. And we are trying to incorporate the most important stem rust and leaf rust resistant genes and the yellow rust resistant genes into the Indian wheat cultivars, so that we can combat these rust diseases. Coming to the history of rust resistance breeding, this breeding program was initiated in 1934, way back before the independence. And at IARI, that's Indian Agriculture Research Institute, the variety NP809 has been released, which was found to have resistance to all the three rusts. Later, the systematic work of how we can analyze the races which are present at present, and the varieties we have, how these two are interacting, that was based on that the systematic work has started in 1931 in way back. And since 1967, All India Coordinated Wheat and Barley Improvement Program has been initiated at New Delhi. Later it has shifted to Karnal. And after the inception of this All India Coordinated Program, a perfect strategic plan for race analysis, varietal analysis, and disease surveillance, how much of the disease is prevalent in which areas, has been undertaken. And just I would like to explain, like the work on rust resistance was started in 3 stages. In the first stage, where we were not having any resistant stocks for hybridization, they were only introducing these resistant varieties from abroad. And in the second stage, the attention was devoted to the production of the strains which are having resistance to individual rust. Like this variety is resistant to stem rust, this variety is resistant to leaf rust, and this variety is resistant to stripe rust. But in the third stage, where we are now, we are aiming for combination of resistance to all the rust, along with the other diseases like powdery mildew, leaf blight, kernel bunt, everything. This is a gene deployment that has been done in wheat growing zones of India. In the table, I have listed the genes that are present in the varieties already that are deployed in zone wise. And on the right hand side, I have presented the genes that are being incorporated into the varieties which we are lacking at present, so that the balance between these genes can help us to combat this rust in the further future also. And coming to the wheat varieties that are being developed at ICAR, Indian Agriculture Research Institute, Regional Station Indore. As I have, as sir told, I was in wheat breeding station for 10 years, and we had a mandate to produce rust-resistant varieties that can be successfully grown in central part of India. And we were working on 2 wheat species, that is bread wheat and durum wheat. And dicoccum wheat was grown only in peninsula, so we were not having any mandate for that crop. And the central India, particularly Indore, it's a hotspot for drought, heat, and stem rust. It means that you need to have varieties which are resistant to stem rust, leaf rust, along with they should be having high yield even when there is a problem of heat or drought in the region. These are the rust varieties which were developed from our station. From 2001 to 2015, we have developed 8 wheat varieties, out of which 5 were bread wheat and 3 were durum wheat. And during the last 7 years, that is after 2015, we have developed 14 wheat varieties, of both bread wheat and durum wheat. Just I would like to explain after 2015 what the varieties we have developed. First, I will be explaining the durum wheat varieties, because the central India that where we are now, particularly in Madhya Pradesh, we have an importance of both growing durum wheat and bread wheat. We are the lead producing state. That's Madhya Pradesh is the leader in durum wheat production, and we have facilities for exporting this durum wheat also, because we have the grain that is produced in Madhya Pradesh, it's having a very good quality and shining, and it attracts the exporters also. The variety is the HI8737 Pusa Anmol. It was released in 2015 for central zone, that's Madhya Pradesh, Gujarat and Chhattisgarh. It can be grown high fertility. It means we can give 4 to 5 irrigations, and timely sown conditions. It means that you need to grow it in November. And it is a medium tall variety. And the genes that are postulated to be present in this variety are SR9E, LR23 and YR2. This is Pusa Tejas, which was released in 2017 with the genes SR2, SR11, LR23 and YR2. And this is the most cultivated durum wheat variety in India. And coming to HI8777, this was released in 2018 with the genes postulated SR7B. And it is released for rain-fed conditions, that you can grow it without irrigation conditions. And the other durum wheat variety is HI8802. It is released for peninsular India, that's Karnataka and Maharashtra, and the genes postulated to be present are SR13 and LR23. HI8805, Pusa wheat 8805, released in 2020 for peninsular India, with the genes postulated are LR13, LR23, SR13 and SR11. And recently released is Pusa Prabhat, released in 2022. It is for central India, that's Madhya Pradesh and Gujarat and Chhattisgarh, for restricted irrigation, that you can give 2 irrigations and you can get good yield. And the genes postulated to be present are SR11 and SR2. Coming to the bread wheat varieties, that are these are used for bread wheat varieties. That mean that they are used for preparation of rotis, roti or chapati, and bread and this one. It is released in 2017, and the genes postulated to be present are SR5, SR11, LR13 and YR2. Like this, we have released many varieties for northeastern plain region, northwestern plain region, and for various conditions, like we can grow some particular varieties in high fertility conditions, low fertility conditions, and also during various durations also. Like this, there are 8 bread wheat varieties that were released from 2015 to 2022. This is the latest variety that was released, Pusa Vakula, with the genes postulated are SR24 and LR24. My intention to show these varieties is, and the genes that are postulated, just I wanted to show that there are varieties available for different conditions, different zones, and the genes, if you observe, we tried our best to deploy the genes that are different from each other, and the genes that are present in durum wheat are different from that of bread wheat, so that even though bread wheat or durum wheat are cultivated together, it doesn't affect the rust resistance pattern, and they will help us to have more resistance to the rust pathogens. Just I would like to conclude and say that this is possible because India is going to give a record wheat production this year of around 113 million tons. And because of this Ukraine and Russian war, we are at a stage that people are very much interested to get the Indian wheat exported, and only because of the collective efforts of plant pathologists and plant breeders in India and abroad also, this has been possible because we have so much record wheat production without any diseases problem this year. And we need to have we need to be vigilant, and we need to know how are the new races that are being emerged, and we should be knowing which are the susceptible varieties that we need to withdraw from the seed indent. We need to withdraw these susceptible varieties, and our breeding program still requires strengthening in prospect of climate change. Like, as you all know that the climate, there is how much effect of the climate change. It also certainly affects the rust pathogen, and the rust pathogen keeps on evolving. So we need to be very much strict to follow our breeding program. And future prospects are like you need to have appropriate management of host resistant genes. Just I have told as you need to know what are the genes present at present and what are the useful genes for the future. and you need to think about durable resistance, that the variety what we have released that should be durable like for 10 years or 15 years, that the pathogen should not attack the variety. And wider replication of genes having minor phenotypic effect, this I could not explain. There are both types of genes, which are major genes and minor genes. We need to focus more on minor genes also, and you need to be you need to have consistent vigilance, and you need to monitor the rust pathogen. And I would like to thank the IPS Academy, Dr. Gupta sir and Chan sir, for giving me this opportunity so that I could share some of the breeding programs that we do it. Thank you so much, sir.